Thank you very much, Claudio, to join this meeting. Yeah. So we try to kind of maximize our visibility of the project, of ALG-AD project, and larger audience. So today I invite to speak with Dr. Claudio Fuentes Grunewald, who is Work Package 1 Technical 1 leader. So Claudio, can you please present yourself and just talk about your role in the project, please?
All right. Well, thank you very much, Ala, to invite me to this new way to communicate our project. This is ALG-AD project. We work with Ala since a long time ago together in the microalgae field, and this ALG-AD project is EU Interreg Northwest project. We are working with several countries, with partners from Belgium, France, Germany, and the UK as well. So in my case, well, as Ala said, my name is Dr. Claudio Fuentes Grunewald. I'm the leader of the WP1, Work Package 1, which is the technical package, and our aim is to basically to run 3 different investments in 3 different countries in the UK, in our case in England, and in southern England, in Devon, in an anaerobic digestion plant. It's called ALG-AD, which is a plant who process mainly food waste from Plymouth. And then we have another investment in France, in Brittany actually, and then we have the other one in Belgium. All the 3 sites work with a different kind of system in terms of the growth system. Some of them they grow in heterotrophic mode. In our case, we grow in autotrophic and mixotrophic, and the Belgian is quite similar to us. So basically, the WP1, the Work Package 1 of this project, the main goal is to advance in this technology because it's quite new. We are doing cutting edge science. Actually, we are in the front line, which is a common word today, but we are in the front line of science. Yes, and my role is try to put together all our knowledge and background and try to be successful doing this work.
Yeah, okay. Thank you, Claudio. Super. So can you please talk a little bit more about the installation and operation of PBI, capacity, and, so how we did this initial steps, just to be on industrial site and basically grow algae there.
Okay. So yeah, it's been hard, but it's been really pleasant too. It's been really challenging, but at the same time, we are learning a lot during this long trip. Actually, we have been working since 2018. By that time, we were planning how to set up a new investment. In our case, we use a autotrophic reactor, right? It's a 7-ton reactor, but the working volume that we normally use is around 5 tons. So we have 5,000 liters of culture. And then we have all the rigs in terms of membrane filtration system, because this is our main system. And then we got the mixotrophic reactor. Right. So basically, if you want, I can explain in detail what is the process that we have been following. Well, so far, what we do, we take the digestate, which is basically one of the waste from the anaerobic digestion process. So this digestate is a really thick sludge, right? And it's full of ammonia and phosphate and all other trace metals as well, but mainly ammonia and phosphate. It have a big content, and actually the concentration of ammonia in our case is as an average is around 5,000 milligrams per kilogram, so which is really high.
Really high.
Yes, and so what we do, we take this digestate from the anaerobic digestion tank, and we pass through a membrane filtration process. In this case, it's a microfiltration. We use a 0.2 micron filter where we separate the sludge, which is basically the waste, from the liquid fraction. And in that liquid fraction, we get most of the ammonia and phosphate, some metals as well, and of course some organic substance. So then what we do, we introduce that digestate, liquid pure filtered digestate, into the photoautotrophic 5,000 liter reactor. All right, we have been doing a lot of tests, so we know now that according to our data, the dilution that we have to use is around 2.5%. That dilution, we get around 180, 190 milligram per liter of ammonia, which is a concentration is high, but the algae can work well, right? So then we do a semi-continuous process. Okay, so basically our growth strategy is to grow in semi-continuous way. So technically, in a weekly basis, we do a harvest, right? In our case, we harvest around 30% of the volume, of the total volume, which is 1.5 cubic meter, right? We harvest from the phototrophic reactor, and we pass through another membrane. In this case, is a clean membrane, all right? And we concentrate that biomass, that microalgae biomass, in order to put in the mixotrophic reactor. So in that mixotrophic reactor, with the concentrated microalgae, we normally concentrate down to 300 liter. So the starting point, for instance, in Chlorella, because we have been using Chlorella and Syndesma.
So two species.
Yeah, two species. So for instance, in Chlorella, we reach around 2 grams per liter when we concentrate, right? And then Chlorella take around 72 hours to reach over 6 grams per liter, right? Okay. And we use a different carbon source. In our case, we are testing glucose, but also we are using other waste. For instance, in our case, we use chewing gum powder in order to add this carbon source. Okay. So after that, finally, what we have done is basically we concentrate this mixotrophic, we harvest the mixotrophic reactor using centrifuge, and we get the paste, right? So in the whole concept is we do the environmental service, which is the uptake of nitrogen and phosphate in the photoautotrophic reactor. We do our new approach, concentrating the biomass using the microfiltration machine, and then we got the mixotrophic reactor where we obtain the biomass that we're going to use later on further in the process in order to test. We're going to do some trials for animal feed. Yeah.
Okay. Claudio, thank you. Just would like to ask if somebody listened to us right now, so you can ask question as well, so we can we'll discuss them as well. But my question is, like in this case, so you said you use Chlorella, and with Syndesma, do you have the same results, or it's slightly different if you use different species?
No, it's quite interesting because in the microalgae biotechnology, most of you probably know that these two green microalgae, they are usable, really useful microalgae in terms of bioremediation. But and actually both of them they are freshwater algae, but they behave in a different manner. So what we found so far, that Chlorella can uptake, for instance, more nitrogen or ammonia in this case than Syndesma, but Syndesma for us is being more robust. So it's more stable, the culture is more stable. Actually, we have been running during nearly a year this reactor, and nearly 8 months of those 12 months, we have been growing Syndesma. So both of them, they're able to grow in photoautotrophic and also in mixotrophic mode. But so far, for us at least, Syndesma is quite more robust and stable rather than Chlorella.
Okay, perfect. Thank you. So another question. So I have just would like to ask about the operation. So we operate the system now more than a year, isn't it? So before the pandemic, COVID, we just have to stop the reactor, if I understand well. So can you explain how this data and what data is recorded and how we will use this data in the project?
Oh yeah. So well, in our case, we have been recording a lot of data, biological data. So basically, how the algae grow, the uptake of ammonium phosphate, the common issues that we have in a higher scale culture. Because you have to keep in mind that we are working in a higher scale. We are working with a culture over 5,000 liters, which is real challenging, and all the abiotic parameter that we normally get in a greenhouse facility. So we got a temperature, we have to deal with cold temperature, we have to deal with hot temperature through the seasons. So this is regarding the technical parameter that we are recording. But also what we have done as well, we have been recording all the data regarding, for instance, energy consumption and water consumption as well, in order to bring this data and to put in a LCA, in a life cycle assessment. Actually, we work with one of our partners is Birmingham University. They are really good in terms of LCA analysis. And another partner that we have in Germany, they're going to do the TEA, so the technical economical assessment. So basically, we have to collect this data, give it to them in order to know if this technology is viable in terms of economic. We know now, and this is quite new to be honest, that we are able technically to grow, uptake, and produce biomass using digestate, right, in a high concentration. But now the challenge is as well is try to use this data, get the LCA done and the TA done, and then try to give this data, this really good data for the stakeholders. So for instance, we know that in the EU, in Europe, in the UK, we got nearly 70,000 AD facilities throughout the whole European Union. So this data will be really useful for them, because we have to remember that in this project, the main idea is to test the technology in order to deal with the nitrogen excess, from the digestate, because we need to avoid eutrophication. Because normally what the people have done, the AD people, they try this digestate and then they spread over the fields. But with this high concentration in nitrogen, we got eutrophication problem. So the idea is to deal with that using this. We are testing this technology, and so far we have given really good result, in my understanding. Yes.
Yeah. So like you said, then avoiding pollution and just keep the environment in a certain level of eutrophication. So this is the main objective, just and we're working on this ALG-AD project in terms of circular economy.
Yes.
Claudio, I have a question from audience.
Yes.
So Siva Subramanian from India, asking question, why we're not using a mixture, so consortium of species instead of single culture. Yeah, and just I'll finish this question. And have you done a preliminary screening of several species before we finalize Chlorella and Syndesma?
Yes, well, thank you very much for the question. It's a really good question, actually. Yes, we have tried consortia, and actually what we have done at the very beginning of the project in 2018, we went down to Langage, to our AD facility, and we have collected a sample of microalgae next to the anaerobic digestion tank. Because when they have to handle this digestate, they connect the lorries, they connect a hose into the tank. So there is a leak in there, and we collect from that pool a sample of microalgae. And in that microalgae sample, it was a consortium. All right, it was mainly cyanobacteria, but also we had some diatoms and some chlorophysia as well, some green algae. So we have tested those consortia, and they are used to live in this environment where we got basically a lot of ammonia content in there.
Yeah.
But the problem that we got when we put this consortia in a closed system with a higher temperature, because that sample was collected from the environment with a really low temperature in the UK case, so the average here is around 10 degrees. So our system worked in 25 degrees. So there was a bit shocking for them, and
definitely
we weren't able to upscale until a massive amount of water. That was the main problem, and also we have to keep in mind that the consortia can work well, but at the very end, we have to do two services. So we have to uptake the nitrogen and phosphate, but also we have to provide biomass in order to test for feed animals. So we want to do a trial with that. And the problem that we have with the consortia is there is a seasonality. So some time of the year, cyanobacteria is the main microalgae in there, but some other time is diatom. So we don't have a consistent and a reliable source of biomass. That is our main process, and that is why we swap from this consortia to microalgae, specifically Chlorella, and to Syndesma as well. And the good thing is that with these two algae, we are able to grow to the two service, but also, and this is really, really good, we have been working without no visible contamination, which is pretty good. So we know that the macromolecule composition, in terms of protein, lipids, and carbohydrate, is quite stable through the year.
Yeah, Claudio, thank you. I think so. This is really open, just like you opened this question really nicely. Also, I would like to add from myself, as well, then we're using this biomass for the animal feed, so we need to have predictable species, isn't it, for the future development. So by regulation, it's another set of the rules for us. Okay, so time is going. Claudio, my question is, when do you think we will restart the operation, and what's your plan for these facilities, in terms of project outputs and deliverables?
Yes, well, as everyone in the world, here in the UK, we're in a lockdown due COVID-19 issues. So we had to stop our operation. We have to stop since the 23rd of March. Luckily, we ran 1 year of this PBR, so we got plenty of data, which is good. Because one of the objective was to test in different seasons through the year. So we got all this data. And our idea, it's always depending of government, government
regulation,
regulation. So basically what we have to weigh is for them, when they say that we can, or the UK can start working again in terms of non-essential work. So technically, we are going to be able to restart our operation down in language. A good thing about that is that we have been keeping our strain, specifically Syndesma. In this case, we have been keeping in small volume down in language, but also here at Swansea University. So we have a backup in here, which is a 1,000 liter culture here at Swansea University. So as soon as we restart the operation, we're going to be able to move that culture to down to language and to restart. So potentially, hopefully as well, by the end of May, we're going to be in operation again. Otherwise, we need to wait until the government say yes, you can do it, and we're going to do it definitely.
Okay. So, but the main objective to run the facilities is what, to get more biomass, more data?
Yes, because as I have said previously, we have been collecting a lot of data. All right, so that work is being done, so it's okay in order to test the LCA and the TA. But what we have to do now is to provide the biomass for the animal feed trial. So this feed trial is going to be performed in France, in one of the biggest pig companies producer. All right. So this is based in Brittany. Is a company that produce millions and millions of pig on a yearly basis. We're going to send this biomass to our partners in there, and they're going to use piglets, so small pigs, in order to test different kind of concentration to put in the pellets to feed the animals, all right? And so our target is to at least to deliver between 25 to 30 kilograms of dry biomass by September. So this is a target, and I think we are going to be able to do it if we start May or June. We're going to do it because technically we can produce nearly 3 to 5 kilograms per week. So we should do it.
Okay. All right. I think so. This is all my questions, Claudio. So it's our starting only point. So if the audience will send more questions, we will organize another live kind of discussion around the results and activities of the project. So we would like to thank you, everyone, and thank you very much, Claudio, to come and just to do it with me first. And this video will be available all the time on website, on LinkedIn and other channels as well. So if you have any questions, so please send us in comments. We will try to reply as soon as we will see it.
Yeah, thank you, Ala, for this first interview. And I think if the audience have more questions for us, we are happy. We are doing science, and this is part of our role, try to disseminate our findings and try to get involved the people and engage all the people through in the world. So feel free to contact us and keep on the loop, and we will reply to all your answers. Thank you very much.
Okay, thank you very much. Thanks. Bye-bye.
Thank you. Bye-bye.
Bye.